What if we could edit the very code of life? I mean, not just treat diseases, but actually cure them at their source, by literally rewriting our own DNA. Well, that's not science fiction anymore. It's the incredible power of a technology called CRISPR. And in this explainer, we're going to break down what it is, what it can do, and the massive questions it forces all of us to ask.
But before we dive into all the amazing science, let's just start with a number. $2.2 million. That is the price tag for the very first approved CRISPR therapy. A pretty staggering cost that immediately brings this whole miracle cure thing into very sharp focus.
And that really gets to the heart of what we're exploring today. We have this technology that can perform what are, for all intents and purposes, medical miracles, but it comes at a price that raises these huge questions about who actually gets access to them. It's a story of unbelievable breakthroughs, but also some really immense challenges.
So first up, let's talk about the breakthrough itself. That moment when this tech left the research lab and stepped out into the real world, creating a cure that, not long ago, was completely unimaginable.
Okay, so meet Casgevy. In late 2023, this became the first ever CRISPR-based therapy to get the green light from regulators. It is a functional cure for two really devastating inherited blood disorders, sickle cell disease and beta thalassemia. Now, instead of just managing symptoms for a lifetime, Casgevy goes straight to the root of the problem and edits a patient's own cells to fix it.
And the results, they are just breathtaking. Take a look at the data from the clinical trials. 94% of sickle cell patients were completely free of those agonizingly painful episodes that just define the disease. And 93% of beta thalassemia patients, they no longer needed the lifelong, burdensome blood transfusions they relied on to survive. I mean, this isn't just a small improvement. For these folks, it is a brand new lease on life.
Okay, so how does this miracle technology actually work, right? How can you possibly find one specific gene among billions of letters of DNA and just change it? Well, let's get into the elegant science behind what we're calling the genome's molecular scalpel.
You know, what's so fascinating is where this all came from. It wasn't dreamed up in a lab to cure human disease. It was actually discovered in bacteria. It's a natural, ancient immune system that these microbes use to fight off viruses. They basically keep a little mugshot of the virus's DNA so they can recognize it and chop it up if it ever invades again. It's just a perfect example of how basic, curiosity-driven science can completely change the world.
So scientists, being clever, repurposed this bacterial system into a kind of two-part tool. You can really think of it in three simple steps. First, you have a molecule called a guide RNA. It acts like a biological GPS, homing in on the exact spot in our 3 billion-letter genome that needs a little tweaking. Second, an enzyme, usually one called Cas9, comes in and acts like a pair of molecular scissors, making a super precise cut in the DNA. And third, and this is where the editing magic really happens, the cell's own natural repair systems kick in to fix that break.
Now, scientists can actually control the outcome because the cell has two main ways to patch up that DNA cut. On one hand, you've got a process called NHJE. It's fast, but it's also a little sloppy. It's perfect if you just want to disrupt or knock out a gene that's causing problems. But then there's HDR. This one is way more precise. By giving the cell a new template to work from, scientists can use HDR to actually correct a mutation or even slot in a whole new healthy gene. The strategy they use really just depends on the disease they're trying to fix.
So, Casgevy was just the beginning. Really, it was just the proof of concept. Its success in editing cells outside the body has totally opened the floodgates. And now, the next great frontier is editing genes directly inside the human body.
A perfect example of this is the Brilliance trial. This was a landmark study. It was the very first time a CRISPR therapy was injected directly into a person to edit their cells in vivo, or inside the body. It was targeting a rare form of inherited blindness, and the results were just incredible. 79% of the participants had measurable improvements in their vision. This proved that the concept isn't just possible, it's actually safe and effective.
And here's a crucial point. CRISPR isn't just for curing these rare genetic diseases. It's also becoming a powerful manufacturing tool. It's being used to engineer the next generation of cancer therapies, like CAR-T, to create off-the-shelf treatments from healthy donor cells. Now, this could make one of the most powerful new cancer treatments way faster, cheaper, and more accessible to thousands and thousands of patients.
And this table here just gives you a little snapshot of how fast this field is moving.
We're seeing trials for blood disorders, for blindness, for different kinds of cancer, and we're even seeing more advanced versions of CRISPR, like base editing, which can make even more precise, single-letter changes to the DNA code. The whole pipeline is just exploding with potential, targeting a wider and wider range of human diseases.
Okay, but with this unbelievable power comes immense responsibility and some pretty significant risks. The story of CRISPR is really a double-edged sword. So let's look at that first edge, the practical hurdles of safety and access.
The single biggest safety concern, the thing that keeps scientists up at night, is something called off-target effects. This is basically when those molecular scissors accidentally cut the wrong place in the genome. An unintended snip could mess up a critical gene, or in a worst-case scenario, even switch on a gene that causes cancer. Making sure this tool is absolutely 100% precise is the top priority for everyone in this field.
And then we come right back to that price tag and this profound crisis of global health equity. I mean, look at this chart. While the cure was developed in the US and Europe, a staggering 80% of people with sickle cell live in sub-Saharan Africa. They're in regions with just no possible way to afford or deliver a multi-million-dollar therapy. So we're left with this cure for a disease of the global south that is almost exclusively available to the global north.
And beyond the safety and the cost, there's also a massive logistical challenge. You can think of it as the last mile problem. How do you actually get the CRISPR machinery past the body's immune system and deliver it to the exact right cells, whether that's in the liver or the muscle or the brain? Solving this delivery problem is one of the next huge frontiers for making these therapies widespread.
And that brings us to the second, and you could argue more dangerous, edge of the sword. Because beyond all the practical challenges are the profound ethical questions that this technology forces us, as a society, to finally confront. Now, this right here is probably the most important bright line in the ethics of gene editing, the difference between somatic and germline editing. On the left is somatic editing. That's what Casgevy does. You treat one person, and the genetic changes are not passed on to their kids. But on the right is germline editing, modifying sperm, eggs, or embryos. These changes are heritable. They get passed down through all future generations, permanently altering the human gene pool. And there's a broad international consensus that this is a line we should not cross.
And that leads directly to the slippery slope argument. The fear is that the line between therapy and enhancement is, well, it's pretty blurry. If we can fix a gene that causes a terrible disease, what's to stop us from enhancing a gene that's related to, say, intelligence or athletic ability? This is the debate that quickly leads to conversations about so-called designer babies.
Ultimately, this technology forces us to ask some of the hardest questions imaginable. Who gets to decide what a disease even is? Who gets access to these cures? Do we risk creating a future where society is split into the genetically enhanced and the unenhanced? Are we stumbling towards a new, high-tech version of eugenics, one that's driven not by the state, but by the consumer market? And so, that's where we're left. We have a technology with the power to relieve an unbelievable amount of human suffering. But that very same power could fundamentally change what it means to be human. The science is moving at lightning speed. But the ethics? They are nowhere near settled. The crucial question isn't just what we can do, but what we should do, and where we as a society decide to draw that line.